Hi, can you all hear me? Okay, so yesterday we were looking at questions from week 12. So that kind of summarized whatever was taught in all the 12 weeks. Today we'll be looking at a few questions from every week's lectures that would give you an idea about what all topics were covered, and we can conclude with that. And if you have any specific questions with respect to any specific topics, we can discuss that here today. If there are no questions, I'll start with week one lectures questions. Yeah, so in week one of genetic engineering course, we were looking at the basic differences between eukaryotes and prokaryotes, differences in their protein making machinery, which is the ribosome. We were looking at how each of it has a different set of rRNAs, ribosomal RNAs, a different large subunit and small subunit. We also had a discussion on how at the genome level there are differences with respect to the presence of intron, how membrane-bound organoids are present only in a specific organ, specific classification, and also on the differences between where transcription could occur. In prokaryotes, transcription occurs in there is no demarcation as to whether a nucleus is present or not. The genetic material is present in a region that is called the nucleoid region. So it is there is no compartment, whereas in eukaryotes you have nucleus, and the number of origins of replication, places where DNA replication machinery could start replication of DNA, is single for prokaryotes. Then, with respect to the structure of the cell outside the cell, we were looking at things like mesosome, the capsule, the cell wall, and you also have a few hair-like machineries. The large hair-like machinery that is involved in motility of bacteria is called the flagella, whereas you also have something called as fimbriae, which would be surrounding the cell throughout and not in a specific region that would help the cell to adhere. Yeah, was there? Sorry. Yeah, can you hear me now? Okay. Okay, I'll repeat that again. I was telling that we were looking at difference. We started by studying how organisms are classified into prokaryotes and eukaryotes. Prokaryotes are lesser defined organisms. They do not have a nucleus. Their protein making machinery, which is the ribosome, is also different from the eukaryotic ribosome with respect to the composition of their ribosomal RNA and also their large subunit and the small subunit. We also looked at how there are specific differences at the genome level, like the presence of introns between exons in a eukaryotic genome. So introns are those non-coding regions of non-coding regions that are present within the coding regions in eukaryotic genome. And only eukaryotic organisms can possess membrane-bound organelles. And with respect to processes like transcription, since the DNA is confined within a particular compartment called the nucleus in eukaryotes, transcription occurs in nucleus and the mRNA gets out of the nucleus and then translation happens outside the nucleus for eukaryotes. But in prokaryotes, you don't have this demarcation and your DNA is present in a region that can be called as the nucleolus. Sorry, the nucleoid region, not the nucleolus, the nucleoid region. And with respect to how replication happens or where replication happens, it is called an origin of replication. Prokaryotes do not possess more than one origin of replication for a closed circular DNA, whereas in eukaryotes you have multiple regions where DNA replication could start. Then we were looking at the different structures that could possibly be present in bacteria. One is the mesosome. This is involved in many processes like formation of cell wall, antibiotic resistance, and so on that has been listed here. So this is different from the capsule, which is made up of polysaccharide and it is attached to the cell wall, which helps in bacterial adhesion to surfaces. Then you also have other structures like the flagella, which will give the bacteria motility. Then moving on, we looked at how plant cells are different from animal cells and how this means that they need specific organelles to process specific metabolites. And one such specific organelle is the glyoxysome, which is only found in plant cells. These are specialized type of plant peroxisomes that contains this glyoxylate cycle enzymes to convert lipid to sugar in the early stage of germination. Then we looked at the genome organization, whether genome organization in eukaryotes. So the genome organization in eukaryotes is based on this amount of strain that the DNA can withhold. So this is one is called the euchromatin or the true chromatin. Here, this is transcriptionally active. It is not densely condensed, which means that it, the transcription machinery can access this, bind to this, and allow for transcription. The other kind of chromatin, can anyone mention the name of the other kind of chromatin? What is the other kind of chromatin? One is euchromatin. What's the other kind of chromatin?
Heterochromatin.
Heterochromatin. Okay, so that is usually condensed. It is bound with this DNA is bound with histone proteins that would allow specific transcription at specific cellular signals only. So that is darkly stained and transcriptionally inactive. Then we also looked at specific organelles like the mitochondria. Here they show the cross section of a mitochondria. So mitochondria is one of the very few organelles that have an inherent DNA within them. Does anyone know if there are any other organelles that can also have DNA within them
Mitochondria,
other than mitochondria?
chloroplast.
Chloroplast.
Okay, right. And do you know how these possibly inherited a DNA? Any idea about that?
Um, Due to evolution.
That is a very, very broad answer. Of course, you can say something happened due to evolution for any question that we discuss. But do you think there is some association with some other organism, possibly in the past, which is not there anymore?
Bacteria, bacteria which I mean does photosynthesis. Maybe that
Okay, does your mitochondria do photosynthesis or your chloroplast does photosynthesis?
chloroplast.
Right. So whatever answer you're saying could possibly reflect that, where cells, so there was a host cell which allowed another kind of cell to stay within itself and then grow. And later on, over the course of evolution, the other cell, which was staying within the host cell, transformed into an organelle. That is one possible explanation why you have these DNA specifically in these organelles. So we were looking at these different, organelles, and we had also discussed on the functions of specific organelles and the classifications. Then we went on to and discuss how photosynthesis happens, where we see in the presence of light how sugar gets converted, and you have these separate photosystems, photosystem 1, photosystem 2, that have their own unique functions. And then looking on other major organelles, we discussed about endoplasmic reticulum. Here, lipid synthesis would happen and conversion of lipid into other pos uh, lipid-based molecules would happen. And apart from that, protein sorting would happen. You have this network of Golgi, which would allow for translocation. But before translocation, you need to decide which protein has to go to which region, for which this endoplasmic reticulum is playing a role. Then we also looked at the other kind of organelle that is needed to deal with toxicity within the cell, which happens by engulfing the bacteria. So this is done by a phagolysosome. And here we have a representation of what we earlier discussed. So your rough endoplasmic reticulum has ribosomes, Your smooth endoplasmic reticulum does not have ribosomes. Because ribosomes are present in rough endoplasmic reticulum, they could synthesize proteins. This is transferred to the Golgi complex, and then the Golgi complex, from the Golgi complex, the route which the protein would take further is decided. So this kind of tells you the role that endoplasmic reticulum Golgi complex would take part with respect to protein sorting. And then, because we were discussing about this phagocytosis, we also discussed about what is the possible condition in which phagocytosis is effective. In order to be effective for phagocytosis or to kill any cells, you need to have an acidic pH. Then we moved on from organelles to separation of the or the division of bacteria into different layers based on the presence of an cell membrane and an internal membrane, which would give you a region of periplasmic space. Now, this periplasmic space is for usually proteins are expressed in cytosol. But if you have a protein that is toxic to the cell, expression of this protein in cytosol should be toxic to the cells itself. And if it is toxic to the cells itself, the cells would die and the cells would no longer proliferate. So to prevent that, what one would do is put a secretory tag with the protein such that this network of endoplasmic reticulum and Golgi bodies would transfer the protein to periplasmic space so it is not present in the cytosol and it wouldn't affect the cell. So once this could be done, what needs to be done is you need to break open the cell specifically so that you do not denature the proteins, for which you have a specific method which you do by giving it an osmotic shock, where you change the concentration of sucrose, thereby giving it some pressure and allowing the periplasmic space to get disrupted and the proteins to get released. This is one method of lysis. The other methods of lysis include sonication, homogenizer, and French press. We would be discussing about a few of these in the subsequent week questions when we discuss that. Yeah, so this is done. Next, next we looked at different pathways that are present in this uh, in general in organisms, which also included PC cycle glycolysis. We looked at specific enzymes, their functions, what is being converted to what, if an enzyme would require a cofactor for its functioning, and how ATP is produced. We also looked at the count of this. 1 minute. We also looked at the amount of ATP that would be produced in total at the end of this cycle. But even to start with, for this process, one needs to have an uptake of glucose. and we saw that this would happen with the presence of GLUT, which is a glucose transporter, and this can regulate glycolysis. So, if insulin binds to the receptor on the cell membrane, then this receptor will promote recruitment of the glucose transporter from the intracellular pool to the cell membrane. So your glucose transporter, which is initially bound to the vesicle, would then move towards the cell membrane. The glucose transporter would fuse and allow for glucose transporter to start functioning. Then it would increase uptake of glucose within the cell. Now, when insulin level decreases, this glucose transporter will move from the cell membrane to the intracellular storage pool, which means that in the same way as to how, based with the help of this lipid vesicle, it was able to come from the intracellular pool and then fuse to the membrane, it would fuse inwards and it would uh, it would take up the glucose transporter. So this is the process by which it can be regulated with in the presence of insulin. Then we also looked at other regulation levels, one of which is in the presence of in different concentrations of glucose levels, how this pyruvate kinase is either phosphorylated or dephosphorylated. This converts phosphoryl pyruvate to pyruvate, which is one important step that would also release ATP. Yeah, then we looked at the different steps that are involved in the Krebs cycle and the role of a pH indicator in media and how one would do a conversion when one has to use the spectrophospher counting chamber. We had an example of the same, and we discussed about how one needs to have make use of this conversion factor to be able to uh, calculate the final number of cells or the cell count. Then we started discussing the dynamics of bacterial growth as to how cells would go from the initial lag phase to the log phase, which is the exponential phase, where cells would have maximal growth or maximal cells will be metabolically active. Then it will be the stationary phase. It follows a sigmoid pattern. After the stationary phase, it would go to the death phase. Then we also looked at the time that it would take for cells, the mathematics behind these dynamics, and methods to measure the OD in order to correlate the dynamics. One method that we discussed was the turbidimetric method, where you measure the OD at 600 nanometers. Does anyone remember why it is 600 nanometers? Okay, we use it at 600 nanometers because at 600 nanometers we have very little absorption from proteins, lipids, nucleic acids, and anything that could contribute to the background noise. So we can specifically look at contribution based on the cells alone. Then in week 3, we were looking about how to extract nucleic acids and the chemistry behind that. We looked at methods like phenochloroform extraction, where we discussed about the biochemistry. So when you have a mixture of protein and nucleic acid, uh, initially that is present in the aqueous solution. So what would happen is the protein will be folded well, and in the folded protein, your hydrophilic side is exposed to the water, and the hydrophobic side is present in the core. But when phenol is added, this would flip. So your hydrophobic side is now present outside, and the hydrophilic side is present inside. So you see a flip. And now by you have because you have inverted, you will be able to separate them based on density. You add chloroform that would help in denaturation of the protein, and when you centrifuge the solution you will get these two layers, an aqueous layer and what is the other layer? Does anyone remember? Okay, so the other layer is the organic layer, which contains phenol and chloroform. Then we also looked at what are the different kinds of nucleic acid vectors that we have. One that was discussed here is the yeast artificial chromosome, which is a vector for eukaryotic nucleic acid segments. We also looked at how to isolate genomic DNA, where ethanol precipitation of DNA was discussed. So here we had discussed that as in a solution containing the nucleic acid, if you put cations, the negatively charged phosphate backbone would interact with the cations, and now the nucleic acids are more or less having null charge. So they would dissolve in water, they will not dissolve in water, and they would precipitate out of the solution. Now, by adding ethanol, it would improve the interaction with the salt. So, this was discussed. Then we looked at specific methods, the differences between the methods. The proteome is the method where you take a look at all the different proteins that are present within a cell at a particular condition. Transcriptome is where you take a look at all the transcripts, that is the messenger RNA that will be produced within the cell. Then we looked at specific methods for DNA, cDNA synthesis, and to be able to get information from transcriptomics technologies. One such thing is, is where first, if you get an mRNA in an eukaryotic cell, you would have something called as a poly A tail. We can make use of this poly A tail to synthesize our cDNA. Now, what one would do is they would take a poly T primer that would anneal to this poly A tail. And now, if you add a reverse transcriptase enzyme, that would make it double stranded and it would create an mRNA cDNA hybrid. Once this mRNA cDNA hybrid is made, you can now use the cDNA hybrid having this poly T tail, and you can use a terminal deoxynucleotide transferase to add a poly C tail. Now, this we will use another poly G primer to be able to synthesize the corresponding complementary strand for this cDNA, with which we'll get the double-stranded cDNA. Then we looked at the different phases of cell cycle and also about PCR, where initially you have two strands and then you have primers. Primers would anneal to their respective sites, and start extending. In the process, the primers get consumed. Once this happens, now the newly synthesized strand can also act as a template, and you would get, after 20 to 30 cycles, millions of copies of your DNA. And we also looked at how in the DNA replication process, the replication fork, many proteins play a role. We looked at the role of helicases that would separate two strands, single-stranded binding proteins that would prevent re-annealing, primase which will give you the initial RNA so that your DNA polymerase can sit on that and start to polymerize, and topoisomerase that would prevent the torsion by DNA breaks. Then we also discussed on how to calculate the TM. There were two methods that were described in the video, but the formula that we usually use is, does anyone remember the formula that we usually use? Does anyone remember the formula that we usually use to calculate the melting temperature, TM?
Two of A plus T and plus four of G plus C.
Right. Another complicated formula was also uh, discussed in that lecture, but this formula itself would do for your calculation. Yeah. So with this, we ended week three's lectures, and then week four. So in week four's lecture, we were looking about a few aspects with respect to RDNA technology, where we discussed about the different kinds of enzymes that we use. Exonucleases are those that cleave from the 5' end to the 3' end. Restriction endonucleases are those nucleases that can recognize a specific site and cleave either at that site or away from the site, depending on the type of the restriction endonuclease. There are four types. We might possibly discuss that. Then alkaline phosphatase is the one that can remove a 5' phosphate and convert that into a 5' OH group. Whereas DNA ligase is the one which would ligate a 5' phosphate with a 3' OH. So we specifically discussed the case of an alkaline phosphatase and how it is helpful in cloning. So here, if we digest a plasmid backbone, a vector backbone with two enzymes, it would lead us to have a 5' phosphate and 3' OH and a corresponding on both ends. So even if the sequences are not very identical, if they are close by, it might lead to self-ligation if the ligase is present. So in order to reduce this false positive, what one would do is it treat it with phosphatase. Now the 5' phosphate which was initially present here is converted to 5' OH. And your DNA insert also treated with the same set of enzymes. So your DNA insert would now have a 5' phosphate on both sides and 3' OH. And in the regions where your 5' phosphate might interact with your 3' OH, your DNA ligase would make it covalent. In the other side, where you have a 3' OH and the 5' OH, The cell's machinery with the replication machinery within the cell would repair this. So this is the role of alkaline phosphatase with respect to cloning. Then we look the role of specific enzyme eco R1 methylase, which would add a methyl group to the A position A G in its recognition set GAATTC. It adds a methyl group to the second A, and if it's methylated, the DNA will not be cleaved. Then we looked about the classification. There are four types: type 1, type 2, type 3, and type 4. Type 1 would recognize a site and cleave 1000 base pairs away. Type 2 will recognize a site and cleave in the same region. Type 3 will recognize a site and cleave some 24 to 26 base pairs away. Then we looked about this one enzyme that would cut methylated DNA called DPN1. So the role of this enzyme with respect to cloning is that, let us say you want to cleave. Okay, this enzyme is particularly useful when people try to make point mutations uh, within an insert that is present inside a plasmid. So what one would do is they would design primers in such a way the primers are fully overlapping the region where they want a mutation, and they would do a PCR to generate the entire plasmid itself. So now your PCR has an initial template that was from the bacteria, which is methylated. But all the other subsequent products are not methylated and are generated in the PCR reaction itself. So to remove this original template where you do not have the mutation from all the PCR derived plasmids which have the mutation, one would use this DPN1 and digest this methylated plasmid. Then specific specifications about sites of the restriction enzymes and ratios for ligation were discussed. Then an interesting concept about whether a particular vector can replicate in both types of bacteria, gram positive and gram negative, were discussed. So this would depend on the origin of replication, and if the origin of replication is compatible to both types, it can survive and replicate. We also looked at the different forms of DNA, which is supercoiled DNA, open circular DNA, linear DNA. All of these have different varying abilities on an agarose cell. Then we looked about how this PBR322 plasmid was made using gene ampicillin resistance from RSF2124, tetracycline resistance from PSC101, and origin of replication from PNB1. Then we looked at a technique called blue-white screening. So this is based on the activity of an enzyme called black Z that would code for beta-galactosidase. So beta-galactosidase can naturally cleave beta-galactoside, but if you give a chromogenic substrate called XCAL, that can also be cleaved to produce an indolil compound that will give a blue color. This technique has been used is is used specifically to identify whether the LAC-Z gene is disrupted in a particular case or not, which would allow someone to identify if their gene of interest has been placed in specific vectors. Then we looked about the bacteriophage lambda and how it has sticky ends that would help it to make attain a circular form. We also looked at other specific vectors like the EMBL3 vector, where you have a central stuffer region between your right arm and the left arm, which will be replaced with your gene of interest. Then we looked at specific resistance markers, like for the tetracycline resistance, it codes for a protein that would the tetracycline binds to the A site of your ribosome machinery. By coding for an tetracycline resistance gene, you would protect your ribosome if tetracycline is present. Then we looked about making E. coli cells competent, which means that allowing them to take up foreign DNA or a DNA of our interest, for which we would play around with the environment of the cell. We would have more of divalent cations like calcium in the environment of the cell that would help in reducing the repulsion between the DNA and the negatively charged membrane of the cell. And if you give it a heat shock, this pore would open, allowing this neutralized DNA to enter, and giving it a cold shock will close the pore. This way, we can ensure that DNA can enter the cell. And we also looked at specific steps like taking cells at the log phase so that they are uh, metabolically active, and when you after adding DNA, if you give it a revival of 1 hour, it would grow for at least 3 generations so that you have more cells that can be plated and would give you your desired number of colonies. Then we looked at how to calculate the transformation efficiency, and also looked at which kind of DNA has the highest transformation efficiency. So we know the differences based on mobility, but what one needs to understand with respect to transformation is that this DNA is it should have an origin of replication. That's the first condition. The second condition is it should be closed circular. Only if it's closed circular, if it the DNA replication machinery can start and finish in the entire DNA. Otherwise, it will be difficult. And even if it is being replicated within the cell because origin of replication is being identified, still your plasmid will not be able to encode for any protein because it would get degraded because it's just a double stranded DNA or linear DNA and not circular. Then we looked at how to make these cells competent. So for this, people use single-stranded DNA that would allow the non-specific sites on the cell wall to be blocked so that your desired DNA can alone enter the cell. And for this, they use salmon sperm-derived single-stranded DNA. Then we looked at an enzyme called zymolase, which will help in digesting the cell wall of yeast and making it a spiroplast. And a picture of the same is shown here. Then for making mammalian cells competent, we looked at a method called the calcium phosphate method and the usage of other methods other than transformation, which is transduction or transfection, bacterfection, and all these methods for mammalian cells were discussed. So transfection is when you make pores on the membrane and give it an electric shock such that it can take up the DNA. In conjugation, you have a sex pilus that would allow for transfer from one cell within another cell. In transduction, a an actual virus particle would inject its DNA within the cell, and in transformation, we would allow the plasmid with a heat shock or chemical methods. Then, with respect to plants, we looked at this organism called Agrobacterium tumefaciens, which would cause a tumor. So, this encodes for a TA plasmid, where we put our DNA of interest and transform the Agrobacterium tumefaciens within the plant cells, and that way we will be able to cultivate it. And like we discussed about beta gal assay, where beta gal, if beta gal gene is disrupted, then giving it the X-cal substrate will not give us the blue coloration. That was also discussed. It's the name of that mechanism is insertion and activation mechanism. Then we looked at the lambda phage, which has a like a CI repressor and a CRO. So here it's a tetramer of dimers. We see that this is mutually, both of it has a feedback mechanism. You have the lambda repressor and the CRO, both of it playing physiological roles. One would do that in a particular growth stage called lysogeny, where the bacterial cells that are being infected with the virus, they would not die, but their machinery is used for making more of the virus. In the lytic phase, what would happen is the bacterial cells would burst, releasing the virus into the environment. And this CI repressor and the CRO repressor are um, essentially involved in controlling this particular mechanism and regulating when to attain this switch. Then we looked at URA3 mediated selection, where the URA3 encodes for an OMP case that would convert a 5-fluorouric acid into a toxic compound, making the cell dead. And how this can be used for identifying whether cells have whether gene has been placed in place of ODCAs or not. Then we looked at sequencing methods. A method that we looked at is Sanger sequencing, where we would have instead of deoxynucleotide, we would have a dideoxynucleotide that will not continue propagation but would rather terminate. So based on the length at which termination happens, sequence the identity of the nucleotide could be understood, and by reverse tracking, one would be able to get the sequence of the gene that we tried to sequence. We also looked at so for week 6 we started by looking at the transcription and the translation machinery. So here the differences in the start codon, the stop codon, all of that was discussed. We also looked at what is the specific requirement for an RNA polymerase to sit or a ribosome to sit. You have a ribosome binding site called the RBS that would help you in translation. We also looked at specific plasmids. Plasmids that we looked at in this case was P-lyse S plasmid that would encode for the T7 lysozyme, allowing reduction in lysozyme levels, reduction in T7 RNA polymerase levels, so that expression of protein is reduced. Then we looked at a few chemical methods with which we can either break open the cells, break open the proteins, or identify specific identify the presence of specific bonds in proteins. So we looked at CNBR, which would convert methine to methionine to homoserine and homoserine lactone, a specific um, recognition site for the factor 10A protease, and for trypsin, it's either lysine or arginine. We also looked at different types of yeast, methylotrophic, those that would survive on methanol, and not non-methylotrophic yeast, and how this can be used for targeted metabolic engineering. And we looked at expression systems in yeast, those that can be naturally expressed and those that need an inducer. So many of these were methanol specific promoters like the MOX or other ones. Then they had also shared a site where you can look at the gene encoding for this and how it was characterized. It's called IGEM. The part number for that particular gene of the genomic region is given here. And then we also looked about this one particular cell line that was derived from macrophages, and information about that is given on this particular website that is given as a link here in the PPT notes. Slide number 20. It's a site called ATCC from which one can order cell lines. Then we looked at how to produce proteins. We looked at the concept of inducing proteins by providing it with an inducer in an inducible system. And in order for the inducer to work, the cells need to be present in a media that does not contain the inducer initially, but only after it grows and reaches a particular OD, it needs to be induced so that protein expression can happen to maximal level. Then we also looked at how a particular promoter helps in maximizing expression. The one that we're discussing here is the polyhedron promoter, which encodes for a particular gene that would be maximally expressed in baculoviruses. Um, so by using that promoter, it would maximize the expression of our gene of interest as well. So we had looked at the logic behind choosing this polyhedron uh, as the promoter. Then we started discussing about protein purification methods. We looked at a method of affinity where using a hexahistidine tag one can separate proteins by using divalent nickel, divalent nitrile or triacetic acid bead. You can either use nickel Na or cobalt Na. Both of it would work. Both of it has the same geometry to be able to bind to a hexahistidine tag. So that was discussed, and also about the transcription, entering how a particular kind of DNA, whether it's nicked, circular, supercoiled, linear, how that will have independent um, uh, efficiency level with respect to entering the cell and surviving within the cell. That was also discussed. Then we looked at In week 7, we looked at methods of disruption with respect to plant cells, where a method called alkali treatment is used to remove particular layers in the cell wall, and how um, detergents like digitonin and Triton X100 would specifically permeabilize either only at the cell membrane or the cell membrane and the nucleus as well. Then we also looked at the osmotic shock method, which was initially discussed in week 2 to break open proteins that are present, break open the cells such that proteins that are specifically present in periplasm can be extracted. We looked at the physical basis of this osmotic shock, where differences in the concentration of the solute outside the cell and inside the cell would lead to rupture of the cell. Then we looked at specifics about sonication, what is the frequency that is needed to lyse cell, and why should it be done on ice? Because proteins can denature at temperatures higher than 4 degrees for extended periods of time. Then we looked at other methods like the bead mill method, which has its own inherent dynamics that can be mathematically modeled, and also looked at how the pressure in a homogenizer can be quantified. Then we looked at an interesting concept called distribution coefficient, as to whether a given solution or the given substance would be choosing to enter into a different phase or be contained within the same phase itself. This is called the distribution coefficient, which would determine whether a particular substance can pollute at different time points if it has a mixture or not. So this is exploited in column chromatography. Now, this would depend on a few factors. One could be the surface area, the other could be the charge on the molecule. The ones that we are discussing as analytes here are majorly proteins. So proteins have differences in uh, molecular weight, which can be used to elute them in size exclusion chromatography, and differences in charge that can be used to allow them to bind to cation exchange or anion exchange columns as well. Then we looked at calculation of this distribution planes, which would depend on the length of the column and the height of the distribution plane itself. Then we looked at the different chromatographic techniques for protein purification, including gel filtration chromatography. Then we looked at how, with variation in the pH of the buffer, the charge on the protein would vary, leading to specific elution. For example, if the pI of the protein is lesser than 6 and the pH of the solution is 8, then the protein would take a negative charge, and anion exchange chromatography can be used, and by using different concentrations of NaCl, specific protein that we are interested in can alone be eluted. Then we looked at what kind of molecules make up this anion exchanger. So in order to be an anion exchanger, it has to be positively charged, meaning it needs to have an amino group. We also discussed this concept of salting and unsalting out, where different concentrations of salt would either break the interactions between the protein and increase their solubility, or in a very high concentration, let they would form a sphere around the proteins, and only the protein-protein interaction would happen that would reduce the solubility and it would aggregate. This biophysical knowledge was later on used to think about proteins better. So in week 8, we looked at how, just a minute. Yeah, so in week 8, we looked at how proteins could move in a gel filtration column, what the void volume is, and what would, and if at all proteins would move out of void volume or not. The answer is actually no, because void volume is meant for proteins or molecules that are really large. Unless your protein is that large, it wouldn't go, and it would prefer to go into these small pores that are present inside the beads. We also learned to calculate the KD value, which is the distribution coefficient based on the elution volume and the void volume. we discussed this concept of radius of gyration, which would allow one to assume proteins as molecules of a particular shape and allow to correlate the molecular weight based on the radius of gyration. So here, this particular idea of correlating radius of gyration with molecular weight based on the shape of the molecule with A is equal to 0.33 for spherical molecules was introduced, which correlates well with proteins. Then we looked at different methods to visualize proteins that could be purified. One method is the STS page method, which is a denaturing condition where the quaternary structure quaternary structure of proteins is when the quaternary structure of proteins is not maintained. Quaternary structure of proteins is lost, because of which you only have the tertiary structure, and the non-covalent interactions are lost. So your protein that would come out here would be monomeric in nature, and to get the good oligomeric status, one would have to run the protein in a native page. By running it on a native page, you would get differences and the quaternary structure would be preserved. Preserving this quaternary structure would give you its actual molecular weight. Then we looked at how urea specifically with this structure could disrupt proteins. We looked at possibly how urea can make hydrogen bonds because it has the same carboxyl group which is present in the C terminal of amino acid and amine groups, that is uh, which makes the actual interactions to form your secondary structures like alpha helices and beta sheets. Now when you introduce this urea, what would happen is, and this urea would pretend to appear like amino acid, and hydrogen bonds would be made between amino acids and this urea, breaking the existing interactions that would hold the structure of the protein. Then we looked at protein ligand interactions and how the requirement of a non-covalent interaction is important for protein ligand interaction. Because if the interaction is covalent, then the ligand will be forever bound. But no ligand needs to be forever bound to proteins because these ligands are meant for a specific function at a particular environmental signal. So only in the presence of that particular environment signal, these ligands need to be bound to protein. And that would happen only if non-covalent interactions are present. Then we looked at another method for purifying proteins, which is a GST tag, which is an enzyme substrate, GST and glutathione. So, like coding for a his tag, if we use a GST tag, what would happen is our protein would would be binding to a bead containing glutathione. This glutathione S transferase with our protein would bind to it. Later, if you add a competitive binder called glutathione, that would bind to the glutathione S transferase present here and it would elute our protein. Then we looked at the choices of matrix and how specific chemistry is exploited to bind specific kind of proteins. Then we looked at linkages, how to attach a particular protein to a particular matrix so that the protein can be immobilized. And we also looked at the geometry of nickel and Da based binding of protein and how EDTA can be used to remove nickel ions. Then we also performed a calculation using the formula that was discussed earlier to calculate the distribution coefficient. And earlier we were discussing about something called as the void volume. So later on we also looked at what kind of molecules can enter the void volume. These are extremely large molecules of the order of more than 1 megadalton. One of that is blue dextrin. Then we looked at a method called desalting where the buffer components in which our protein is present is changed to a new buffer. Hmm. Then we looked at different kinds of electrophoresis method. The one that we were looking at in this particular case is the moving boundary electrophoresis, where you don't have a physical medium like a gel, rather you have a solution. And what would happen is upon the application of an electric field, molecules would start migrating. We looked at how we looked at the chemistry of polyacrylamide gels. So here you have two components, an acrylamide and a bisacrylamide. Acrylamide would form your polymers that would appear like straight, that you can imagine like straight lines, and bisacrylamide is the cross-linking agent that would create cross-links between multiple acrylamides, which would give you the mesh-like structure across which, after movement of proteins would stop migrating. Now, this polymerization is the same method that chemists use. This is called free radical polymerization. Here, TEMED and ammonium persulfate are used. So, here we also looked at how to first track whether the STS page is running or not. And once we know that the STS page has finished running, how do we look at our proteins? So to track our whether our STS page is running or not, one needs to use a dye that would co-migrate with your protein, meaning the dye will have the same migration rate of that of our proteins. So when we load the dye with our protein, it will run, but it will be visible on the gel. But this wouldn't stain the gel, so we use specific substances like Kumasi Brilliant Blue that would bind to lysine residues in the protein and make the gel appear clearly with the blue tint. So your protein will be distinct. Then we also discussed about how sodium dodecyl sulfate, which is a detergent that would add a negative charge to every two amino acids within your protein, making your protein linear and denatured, and the charge density in your protein in all the protein samples is now uniform according to its molecular weight. So when your STS page is run, it would run only on the basis of mass and no charge. Then we looked at a few minutiae like adding an organic solver on the solving gel layers top so that the air in the atmosphere doesn't prevent the entry of oxygen, thereby reducing the rate at which polymerization happens, and how the pore size can be manipulated by changing the acrylamide bisacrylamide ratio, and how glycerol can use to increase the density so that your protein which you load into the gel can actually enter the well and not go away with the solution. Then we looked at the concept of how pH plays a specific role in STS page. In STS page we have two gels, stacking gel and a resolving gel. The stacking gel is used to stack all the proteins into a thick thin layer so that there is no differences in the starting migration. Whereas the resolving gel allows you to separate your proteins based on molecular weight. So the stacking gel is made at a pH 6.8. So the protein is now sandwiched between your glycine and chloride ions. As it moves towards pH 8.8, glycine ions are now having better mobility and would move forward, and this is solely due to the deuterionic nature. Then we looked at the concept called urea gradient page, where you have on one axis increasing urea concentration, and perpendicular to that axis is how electrophoresis happens. Now, what would happen is when you do the electrophoresis, you see that the protein would migrate in a particular fashion, and what one can observe when they run a mutant protein and a wild type protein is the particular concentration of urea at which they see a shift. So if there is a reduction in the uh, if there is a difference in the way the protein is migrating as uh, in in the presence of a different concentration of urea, one can correlate it with the folding nature and also the rate at which this unfolding happens that was discussed earlier. We also looked at what vertical streaks are and when this would happen, and how to calculate the molecular weight of a protein oligomer given its molecular weight in the native page and STS page. Then we looked at the experimental setup for Western blot. So in Western blot, we would have a target protein and a primary antibody that would bind to the target protein. Then the secondary antibody would mostly be conjugated with an enzyme in such a way that the secondary antibody would recognize the primary antibody. And if you give the substrate to the enzyme, it would give us a colorimetric or a chemiluminescent signal with which your protein can be detected. Then we looked at another blot, DAB nickel chloride blot. In week 10 class, we were looking at a little bit chemistry of these biomolecules. We started with looking at the electromagnetic spectrum and how there is a difference in the frequency and the wavelength as we move down the scales, and how we use specific regions of the spectrum for characterizing specific features. For example, we use UV visible spectroscopy to characterize the electronic transitions. By electronic transitions, we mean movement of electrons from a particular uh, orbital, bonding sigma, bonding pi, or a non-bonding, to an anti-bonding sigma star and an anti-bonding pi star, and how these can be characterized at specific wavelengths within the UV visible spectra. Then we looked at this concept of correlating absorbance in UV visible spectra to concentration, where first we discussed this topic of transmittance and absorbance and how they are inversely proportional, and the Beer-Lambert's law, which gives you a mathematical equation to quantify absorbance value and concentration based on the length of the path through which the light travels, and molar extinction coefficient. Then we looked at other methods. We didn't go into much detail about the electron paramagnetic resonance spectroscopy, but that would make use of the spin of a molecule or an atom. Then we looked at vibrational spectroscopy that is used to identify functional groups. So depending on the kind of bond and the environment a chemical molecule is present in or possesses, it has many degrees of freedom to rotate and vibrate. This can be exploited to characterize or to identify specific molecules. The An example shown here, CH stretch can be quantified from at 3000 to 2850 centimeter inverse, and so on. Whereas the double bond CH stretches from 3100 to 3000 centimeter inverse, which varies significantly from the C bond H stretch. We also looked at the parts of a spectrophotometer. It would have a monochromator that would um, give it only one wavelength, and then the sample will absorb the light, and that will be detected as the absorbance. We also looked at a few spectroscopic properties like the hypochromic shift, and how it is relevant with respect to nucleic acids. we see that in single-stranded DNA, you have higher absorbance, whereas in double-stranded DNA, you don't have higher absorbance. This is because in double-stranded DNA, your nuclear bases which contribute to the absorbance are hydrogen bonded to each other, so the electronic transitions do not um, happen to that extent. Whereas if your double-stranded DNA is now denatured to single-stranded DNA, that would give rise to more freedom, so the electronic transitions can happen, leading to increase in absorbance. This is called the hyperchromic shift. We also discussed the differences between bathochromic and hypsochromic shift. In bathochromic shift, absorbance happens at a different wavelength, an increase in the wavelength, whereas in hypsochromic shift, the absorbance happens at a lower wavelength. Then we looked at what a chromophore and an oxochrome is. Chromophore is the one that is responsible for um, absorption. An oxochrome facilitates this, and this can either increase or decrease absorption, or it can shift the wavelength as well. That is specific to the chromophore oxochrome pair. Then we looked at how disulfide bonds are formed with respect to in the context of proteins, where you have this cysteine amino acid. So intramolecular and intermolecular disulfide bonds can be formed, but this is reversible. This is reversible if you covalently attach a particular moiety to the thiol groups in cysteine. Having a reducing environment would also make it attain or shift the equilibrium towards making breaking the disulfide bonds, but that's not a permanent solution. Instead, alkylation with iodoacetamide is what would covalently disrupt this, making making the creation of disulfide bonds very difficult or close to impossible. Then we looked at how to calculate the degrees of freedom and specific examples of um, the fingerprint region and so on. Then finally, we also looked at the applications of biotechnology, where we looked at plant and animal biotechnology and how BT toxin play was designed to specifically inhibit Bacillus thuringiensis, uh, the worms that would take up this Bacillus thuringiensis and how that would kill in a specific site. Then we also looked at how herbicides interfere with biosynthesis of metabolites that are essential for the cell, and how this can be um, eliminated by um, how this is specific to only weeds and not the plants of our interest. Then with respect to plant tissue culture, we discussed about different kinds of potency that cells would have: totipotency, pluripotency, multipotency, and so on. We also looked at the differences in the media composition. What is the nitrogen source? What is the carbon source? What is the macronutrient? What is the micronutrient? And how to operate an autoclave and what are the conditions, such as the temperature and the pressure, and what is the necessary sterilization procedure other than autoclave with respect to plants, where you add something called as 2 in 20. So when you add 2 in 20, what it would do is it would reduce the surface tension so that whatever you're going to coat your plants with for surface sterilization would reach all regions of the plant and will not be sticking onto particular regions alone. Then we also looked at uh, how by performing artificial insemination, natural traits are lost. Then we looked at specific cases of the advantages of this genetic engineering technology. One in the production of cheese, where we want to reduce this clumping and cause this aggregate, for which we use an enzyme called the rennet, the structure of which is shown here. Then we looked at how within the body, with respect to particular diseases, by supplementing particular enzymes at the pathway level, certain pathways can be disrupted. And also, we discussed about production of monoclonal antibodies, which is a chimera between myeloma cells and spleen cells, which would give you hybridoma. And their selection would happen in this HAT medium, which makes use of the idea of cells having the ability to create nucleotides. Then this would have the ability to create specific monoclonal antibodies. Yeah, so with this, we have majorly revised all the topics that were discussed earlier for the course of genetic engineering. If you have any specific questions, we can discuss that now. Okay, if there are no more, I'll wait on the call. If there are no more questions, you can leave the class, or if there are any questions, you can ask me.